Welcome back to BIOB 105, Biotechnology. In this lecture, we're going to be talking about Chapter 5, Bacterial Transformation, and we'll be talking about some things that we'll be doing in lab in the upcoming week. So what we'll talk about here is how bacteria acquire genes from outside their genome through transformation, transduction, and conjugation. And we're going to emphasize transformation because that's a procedure that we're going to perform in the lab. We're going to discuss applications of this to what we actually do to put genes into bacteria for the biotechnology industry, and go in detail in how we perform that experiment so you'll understand what goes on in that. So behind everything that we're doing in biotechnology, we are trying to introduce genes into organisms to give them desired characteristics or to produce proteins that have some value. So we've talked about the central dogma or flow of information. So we start with the information in DNA that's transcribed through the process called transcription or copying into RNA, and then RNA is then translated into protein. And once you have those proteins, then you have the particular traits. So we'll be talking about that in the context of what we're going to do with our bacteria in lab then. So general background, bacteria can change. I mean, all organisms change over time. That's the process of evolution. And there's a few definite ways that bacteria can change either slowly or rapidly. So a rather slow way to change in terms of the effects on the organism is through mutations. So mutations change a single base in DNA, and that usually changes one amino acid in a protein, and that may or may not have an effect on the organism. You can also do things like create stop codons that cause the protein to not be produced, which can be devastating, but sometimes changing a single base can give the organism an advantage if it changes the protein some way. A more rapid way for bacteria to change, and one that explains some of the rapid acquisition of antibiotic resistance and other traits, is through movement of whole genes. So remember, an amino acid is just one very small part of a protein. A single base is just one very small part of a DNA molecule. If we can take up segments of DNA, we actually acquire whole new banks of information that give the organism new characteristics. So in transformation, bacteria take up DNA from their surroundings, and I'll describe in a minute how that can happen. Another mechanism that we will talk about briefly is transduction, which is where bacteria take up DNA, or the DNA is moved through the activity of a virus. And the viruses that infect bacteria are called bacteriophages or phages. So they move DNA from one bacteria to another. And then bacteria can transfer directly in some cases through conjugation. And so they literally transfer DNA, and usually these are small segments of DNA, but sometimes part of the chromosome can get transferred, but directly through living bacteria. So both of these mechanisms require either a virus or bacterium to be able to transfer. But transformation is interesting because it's literally DNA that's coming from the surroundings. And where can that happen? Well, maybe bacteria died in the surrounding area, and then that DNA crosses the cell wall and cell membrane and actually gets into the bacteria. So transformation was discovered by Frederick Griffith. There's a little bit of a discussion of that in your chapter, in chapter 5. So we'll just talk about what we do in the laboratory. We know that we can get bacteria to take up this naked DNA, and they have to be what we call confident to do that. So we have to basically permeabilize the membrane and wall so they can take that up. So in nature, these can offer survival advantages. So if they do pick up a resistance gene to antibiotics, it helps them survive. If it's a pathogen that's in the human body, that would enable them to survive the antibiotic. There can also be virulence factors, so things that make something that increase the pathogenicity or deadliness. And there's a few well-known examples of that where bacteria have acquired genes that enable them to produce toxins and things like that. So this was discovered by accident by Frederick Griffith, kind of an interesting story, something we can talk about a little bit later. And later it was discovered that you can artificially induce this in the laboratory. And this has then become a mainstay for cloning genes and enabling us to express those genes in host cells. So the genome of bacteria is pretty small. Most bacteria have around 1 million or so 10 to the 6 is a million. So about 4.6 times 10 to the 6 is the rough genome size of E. coli. It varies a little bit because strains can vary a bit. If you would stretch that DNA out, so this is a nice EM picture of bacteria that's been permeabilized and their chromosome is spreading out all over the EM grid. So we have a cell that's about 1 micrometer or so in length, and we've seen these in the microscope, so they're pretty small. And actually, the chromosome, if you would completely stretch it out, would be 1 millimeter. So it's a very thin molecule. You can see very thread-like, so it has to pack itself into the cell. And that gives us about right around 4,400 genes that enable you to build everything that an E. coli would make in terms of all its proteins. If you compare that to a plasmid, and we kind of think of plasmids as mini chromosomes, they're on the order of a few thousand base pairs, so maybe 1,000 to 100,000 base pairs. And there's just usually a few genes in those plasmids. So the one we're going to look at is pGlow, and that's been sequenced, so we know exactly the length of it. It's 5,371 base pairs. It's a circular molecule, so much smaller. So 5 times 10 to the 3 compared to almost 10 to the 6, so 3 orders of magnitude smaller than the whole genome. So if we were going to draw that here, if this represents the whole genome, my little plasmid would be something like this, so tiny little circle there. So we know there's lots of different plasmids in bacteria in nature. Some of them are what we call fertility plasmids, and these are actually used in bacterial conjugation, where bacteria transmit DNA between cells, and so these are transferred, and some of them just simply code for the ability to be transmitted. It's just a DNA that gets transmitted, and that's all, and then it can transmit to another cell. So really no more function beyond that. What we call R plasmids have resistance genes that are usually for antibiotic resistance. And one example is the one in the pGlow plasmid. It actually has one of those plasmids, so its abbreviation is BLA, which actually stands for beta lactamase. So this is an enzyme that can degrade penicillin or ampicillin. So all of the psyllin antibiotics. So in other words, if the bacteria have this gene and it's active, they can degrade the antibiotic so the antibiotic won't kill them. And that usually stops cell wall synthesis, so it basically can block the activity of that drug then. So one of the things that all plasmids have to have is what we call an origin of replication. So if we're going to copy the chromosome, we have to copy the plasmid. And so they have an origin. And many of these are what we call high copy number. So in a typical bacterial cell, there's only 1 chromosome, but you can look at the number of plasmids, and these can be very high numbers. They could be maybe dozens or hundreds. And that's a real advantage if you want to put a gene into an organism and then get it to express a lot of that protein. If you have more copies of the gene, it's going to make more protein then. So we can represent plasmids in a variety of ways. We can show them as little circles. We can indicate the locations of the genes. So this is pGlow again with its BLA gene, its origin of replication, and I'll talk about these other features here. So that's one way to represent it. We can also show it as a map, and what this map shows is a little more detail about the origin. This shows the direction that the gene is actually transcribed when you're making an RNA. And it also has the restriction sites. So if you were going to clone into this and put other genes, you would know what restriction sites you could use. So these represent then the specific sequences. And even more detailed than if you'd actually show every base pair. That'd be a lot of letters to represent, because it'd be over 5,000 letters, but that's another thing we could represent. If you're looking this in the microscope, so if you look in the electron microscope, this is what the plasmid looks like. It's a circle. A lot of DNA gets highly coiled, so both the chromosome and plasmids get super coiled, so they'll actually fit into the cell. So that's a really interesting feature of all DNA molecules. So how does transformation work? So what essentially has to happen is, if these are the little plasmids, they have to get taken up into the cell. So this represents the chromosome right here, the red line. And then once they're taken up into the cell, then we have to have a way to know that that plasmid is there. So not every cell in the culture is going to take up the plasmid. So how do you differentiate between the ones that have the plasmid and the ones that don't? So if they've taken up the plasmid and they have this beta lactamase genes, what they have to do then is activate that gene, produce the enzymes. That takes a little bit of time. produce the enzyme. And then if you add ampicillin, you're going to kill all the cells that lack the plasmid, and you select for the ones that do have the plasmid. So this is called antibiotic selection. So we kill the cells that didn't get transformed, we select for the ones that did get transformed. And that's a really, really key feature in every transformation experiment and almost anything we want to do. If you want to add a gene, you have to have a way to know that you've added the gene and select for the ones that you've had the gene added to. So that's one part of the plasmid. The other thing is we're going to look for this GFP, or green fluorescent protein. So not only are we going to change the genotype and this ability to resist antibiotics, but we're also going to add a trait that we can actually visualize. And green fluorescent protein was discovered in the jellyfish. So this same basic principle is used in other genes, and I think we've looked at something similar. You can take another gene from another organism. So you could take whatever gene of interest. We've talked about using the insulin gene. We splice those together, we transform cells, and we get it to produce insulin. Well, how do we get it to do that? We need to have this selection, and then we need to have the signals for it to produce the protein. So what's very advantageous and very attractive about the green fluorescent proteins, we can actually see that protein. We can actually look at it under fluorescent light or UV light, and we can get that glowing to occur. And so we can know that the cells are actually producing green fluorescent protein. And if we tag it to another protein, then we can say, well, if it's producing green fluorescent protein, it's also producing this other protein that we can't see maybe by fluorescence. So this is referred to as a reporter gene. If I couple my P-glow plasmid to my green fluorescent protein, it'll sort of report on the fact that that gene is in there and functioning, and then I can actually add other genes to that whole construct then. So this was discovered in the jellyfish, and if I get time in lab, I'll show you a little video of the green fluorescent protein jellyfish, and probably put a link on Eagle Online so you can see that. There's a lot of different organisms that produce fluorescence, but this is one that was well studied. So they were able to clone the gene from that organism and put it into this plasmid, and then study it in E. coli. So we can actually isolate the protein. That's another lab that we're going to do in a couple weeks. We'll actually isolate the protein from the bacteria and be able to see that. So this glowing has been applied to other systems. So essentially the way this works is a reporter or tracer protein. So normally you have a gene. So this is represented here. We have a gene, and under the right signals, we produce a given protein. If we add the fluorescent gene in, then we can actually see the fluorescence, even though we can't maybe see the protein itself. We can see that fluorescence. So that helps us visualize where this activity is taking place. So this has been applied to a lot of organisms. We're going to apply it to bacteria and make glowing colonies. You could put this in a variety of organisms. So you can put it in fish. It's been put into plants. It's been put into almost anything that you might imagine. So it's a visual marker. We can use it to follow biological processes. We can localize genes. We can watch cells move. So this is the zebrafish. It's a common aquarium fish. It reproduces very fast. And it's actually used in the laboratory because their embryos are almost transparent. So one example was they were studying heart development and trying to understand the genes that control when hearts don't develop properly. Like sometimes aorta don't form, and we know these are conditions that occur in humans. So if you can understand the genetic principles for that in a fish, you can actually apply it then to other vertebrates, and including mammals and in humans. So being able to watch cells move very early in the embryo help you trace how the initial cells that are going to form the heart and other blood vessels develop. It's also been applied to looking at nervous system development. So that whole cell fate and development has been really important. So looking at organ formation. And then the whole idea is looking at transgenics. So ones that we put an extra gene in, we can actually identify where that gene is localized. So I'm going to stop at this point, but the next thing we'll talk about is then what we're going to do in lab in the transformation and looking at P-glow in bacteria then.